Hi, thank you for joining us for this webinar. We will be talking about programming life for a better world. We will also be talking about bio-innovation and biostrategy. I have the privilege to be with three great people and great speakers, and we will have a very beautiful afternoon in Spain and morning in the States. I want to say that we are in the beginning of a big wave, a big bio-revolution, which is triggered by technologies that we call synthetic biology. And this is the main topic that is bringing us in Navarra to have a conversation with key people in the world, because even though we are in a small corner in the south of Europe, we really want to be protagonists and we want to partner with the best all over the world. So Paul, I think I met you last year in China and then in the States, and I am a big fan of you. I am your admirer. I want to follow your steps. You have been promoting synthetic biology in different scenarios in the UK and beyond, and I think in every continent. Paul Freemont is the head of the synthetic biology department in Imperial College. He's also the co-founder and director of the Center for Synthetic Biology, and he is the core director of the UK National Innovation and Knowledge Centre for Synthetic Biology, known as Symbicity. He is also leading a global movement to build biofoundries all over the place, and I am hoping that soon we will also be part of this network building a biofoundry in Navarra. So thank you for joining us, Paul, from London, I believe, and you have the screen for yourself.
Thank you very much, my guys. Very nice introduction. Appreciate all your words. And it's very difficult following Andrew sometimes. He's a very inspiring speaker. But I'm going to drill down a little bit and tell you a little bit about our work on biofoundries. So I'm just going to share my screen, and hopefully that will work. Get rid of this one. Okay, and I've got that one, and I share screen. Okay. All right.
Yes,
so
working.
Great, thank you very much. And I just put it onto full screen. So I think, so I'm Paul Freemont, and I'm based at Imperial College in London, although I live out in the countryside actually, so that's where I am now. So my internet is not great, but as Andrew pointed out, I think extraordinary revolution going in biology and biotechnology, and has been for a little while. And I think there's a huge opportunity here for us to sort of reconsider the role of biotechnology in our, I'm going to turn off my video just to give me more bandwidth, to reconsider biotechnology as an opportunity to repurpose many of the things that we take for granted, and including sustainability, bio-based production systems, reducing problems around climate change, bioremediation, and many other things that biology and biotechnology could address. Now, one of the interesting things about that is that synthetic biology, which Andrew described beautifully in the introduction, part of this biorevolution, and then I would add also biofoundries is another part of the revolution. And I'm going to tell you a little bit about biofoundries around designing, automation, prototyping, and production. So what are biofoundries, and why are they a new concept? So if we consider at the core of what Andrew was talking about was this idea of writing DNA, and this is essentially part of this, what we call the synthetic biology design cycle. So this is the design, build, test, learn cycle. So this is where one can consider the systematic design of biological systems by rewriting their genomes, for example, or by writing DNA and putting that into cells that would carry out particular defined human designed functions. And so in this systematic design cycle, you begin to learn, and through this process, you will, using tools like machine learning, artificial intelligence, some statistical methodologies. The ultimate aim is to learn how to build robust and predictable genetic systems that operate in living systems. And so biofoundries facilitate this design, build, test, learn cycle to accelerate the prototyping of these biodesigns for specific applications. So if you think about a lot of our great technologies, they've always had a prototyping environment. So for aerospace, there is the wind tunnel, which actually is still used in certain contexts. For computers, we had the breadboard scenario where things could be designed before implementation. And then in biotechnology, we've really struggled to have what I think is a prototype environment, an environment that allows you to test your genetic designs. And synthetic biology offers this environment through the design, build, test, learn cycle. And also the biofoundries offer this opportunity as a prototyping environment. So we are in the age of digital biology, and this is a slide I took from Synthase, which is a startup company in the UK. And Synthase are very interested in the idea of the design of biological systems. So this is biodesign, biosimulation, using models to actually build up processes that would allow the execution of those designs using experimental methodologies. So it's that connection between the biodesign and the software and the algorithms with the physical implementation of those designs, so that one can get analytical data, which can then feed back into that design cycle. So this idea of design and execution becomes very important. And so biofoundries provide that integrated infrastructure. They provide the infrastructure that allows for that sort of prototyping of genetic designs. So they often have liquid handling robots, integrated bioanalytics, the automation and high throughput workflows for molecular biology. Many of the high throughput DNA assembly processes that Andrew talked about are often carried out in automation platforms now. This could be from plasmids even up to whole chromosomes. High throughput testing of engineered cells and strains of bacteria, and then BioCAD tools, software tools, and integrated ICT workflows, so that the process of design becomes well documented, and that the process of measurement and metrology and standards and characterization all become part of the whole workflow. And this then allows data to be curated properly. It allows tools like machine learning to be applied. And the whole concept is to learn how to do biodesign, basically. And the beauty of these infrastructures is that they can also be accessed via the cloud, and they are a pathway to scale up. So the prototyping environment can often lead into as the scale-up environment. So this connection between the downstream processing and the upstream processing can be carried out within this environment. Now, biofoundries happened early in the synthetic biology startup. So Amyris had one of the first biofoundries in 2003. Ginkgo Bioworks has a very large biofoundry in Boston. And then we have companies like Transcriptic and Zymogen also around this whole concept of automation and biofoundries. Now, the London Biofoundry, which I'm co-director of, which I'll tell you a little bit about now, provides this industrial and academic researchers with access to this suite of robotic analytical equipment, which allows you to prototype your genetic designs and characterize them thoroughly and systematically. So we have a whole suite of automated liquid handling robots and measurement tools that you find in normal molecular biosciences departments and divisions and research organizations. But the beauty is that we integrate all of this around this design, build, test cycle. And therefore, we can carry out things like DNA assembly, high throughput DNA assembly, strain engineering, high throughput strain engineering. We're doing a lot of work on cell-free systems and also analytics and also software developments, to name but a few. Now, the beauty of the biofoundry, it also underpins translation and innovation with expertise and facilities. So our biofoundry has enabled many academic and industrial projects, and particularly quite a lot of startups that we've supported in the synthetic biology space. And so when Juanjo asked me about my talk, he mentioned about biofoundries and bioinnovation. So I'm going to focus on the innovation projects that's come through the biofoundry. And this is by no means all of them. This is just a small selection. this is a project here with a company called Ambimass. And one of the problems when you're doing metabolic engineering is that you have to screen a lot of strains to see if your strain is producing a product at a particular titer, particular yield, whether your strain is robust for that production process as you scale it up into bioprocessing and biomanufacturing. So one of the beauties of this technology is called REAMS, whereas we've been working with this company for a while now, is this side allows the direct analysis of colonies of cultures on a 96 well plate by laser ablation, and allows you to select out colonies at a very early stage in the design cycle that are producing particular products at particular levels, which is a very interesting technology. And as you can see, you can do a very rapid screen in that graph there with the Ambimass technology. The Ambi screen is shown in blue, where you can screen up to 10,000 samples within 12 minutes. Enzbon's an interesting company. It came out of Oxford University, and they're very interested in predicting enzyme activity using artificial intelligence. And they wanted to do this obviously with accuracy and highest throughput. And so we helped them develop a proof of concept to verify their in silico design of enzymes. And we did a lot of high throughput analyses of protein variants. We characterized a lot of the products, a lot of the muted enzymes, and developed a lot of high throughput assays for them. Pure Affinity is another startup company. This one came out of an iGEM team at Imperial College London in 2014. And Pure Affinity that's their strapline: protect the environment and human health by engineering targeted materials to capture chemicals. And the CEO, Henrik Hegemann, was listed in MIT Tech Review as one of the innovators under 35. They have been incredibly successful, and the London Foundry is currently supporting them. And we've been doing performance testing from initial product development to end product testing. We've been helping them with some of their manufacturing processes in terms of optimization, refinement, and we've been providing quite a lot of advice and consultancy for them. Another larger interaction was with GSK. This is a pilot project to produce active molecules of therapeutic interest. They were interested in, could we use high throughput systematic combinatorial DNA assembly and look to see if they could screen for colonies with high product titer and use some of the data around that to do some learning. So we carried out that project, and one of the outputs actually for our perspective was a miniaturized high throughput plasmid DNA library prep for next generation sequencing. That's a very useful output. So this was an interesting project from our perspective. We've also got technology partnerships. This is just one with a company called Analytica Gena. So the foundry is working closely with AJ to evaluate their workflows, evaluate some of their robotic platforms, to implement some of the advanced methods on their platforms, and to also allow startups and SMEs to access those platforms through the BioFoundry for some of their own work. And the idea there is that we can share experiences and developments through journal publications, application notes, etc., and also allow training of third parties on the platforms. We've also recently just pivoted to support the national COVID emergency. I just got one slide on this. So this is the excitement about BioFoundries. You can really pivot them. They're very adaptable infrastructure. So within about 6 to 8 weeks, we ended up repivoting some of our workflows and produced an end-to-end qPCR workflow that could test 1,200 samples in a 12-hour period. It needs a very small area, small lab, and those are all the infrastructures required. And we've now got a paper accepted in Nature Communications on this work. Both platforms are now implemented into hospitals in London, and in fact, one of the hospitals is now expanding to buy 5 of these platforms for their own COVID testing. Now, the Global BioFoundry Alliance is something which I'm really quite passionate about. And we established this last year in May in Kobe in Japan. The Alliance is being established worldwide. It provides an integrated infrastructure. So BioFoundries, as you know, provide this integrated infrastructure to enable this rapid and testing, as I've described. Many countries around the world want to benefit economically from these developments. So there's been a whole investment in public funded biofoundries or non-commercial biofoundries around the world. And so we built the Global Alliance to address this massive growth in non-private biofoundry developments. Currently, we've got 29 members, and you can see on this world map, you'll notice there is a distinct spot missing in Spain, Portugal, and France, which I think we hope to address in the next period, but we have quite a lot of members in Scandinavia and also in Germany and also in the UK and elsewhere around the world. And the great beauty about the BioFoundry Alliance is that it's there to develop and promote and support non-commercial activities. We intensify foundries, developing joint protocols. We have working groups that meet regularly. We have this annual meeting that we run, and we're beginning to develop resources that are going to be open source and free to the whole community. And so, in summary, I think biofoundries are not the whole solution, but they provide part of the solution towards accelerating biotechnology research and innovation. They provide wonderful adaptable infrastructure for prototyping genetic designs for specific applications. They support both academia and industry. They really foster an innovation community around synthetic biology. And of course, for particular national challenges and emergencies, they can pivot into developing workflows for diagnostic testing, for example, as we showed in my own lab. So the Global BioFoundry Alliance is also there to promote the sharing and open source technology. So I think we're just at the start of this journey, as Andrew said, and it's a very exciting journey. Just to thank the amazing team in my lab who worked their socks off to do the COVID-19 testing platform work. And I'll leave it there, and I'll stop sharing my screen and put on my video. I hope I did.
Thank you so much, Paul, for your very complete and inspiring presentation. I hope to be part of this conversation to cover the spot in the south of Europe, and our government and our stakeholders in Navarra are really dreaming of that now. So we hope to make that a reality in the coming years. I also appreciate, and I think it's a good proof of concept, your work that you're now publishing about using the BioFoundry as a prototyping environment to create in record time new tests and to also validate current ones. So that's amazing. And I want to ask you in a few minutes, we had a question about biosafety and biosecurity. When we are working in a community and maybe programming organisms, but maybe we need to partner with a BioFoundry or with other companies. So how can we make sure that all these connections are allowing us to work safely?
Yeah, that's a really important question, and biosecurity has been a major part of the field of synthetic biology since its inception. And I think, so we have a process in our own BioFoundry where every project is vetted by a board, and we look at the proposal, and particularly from industry and companies, and we checked to see whether there are any issues that people have not thought about. So we do have an internal vetting procedure for all the projects that we work on and collaborate on with external parties. Within the universities, we have our own, normal genetic modification regulation, GM regulations. And synthetic biology at this current state fits very nicely into most of the existing GM regulatory frameworks that exist around the world. Now, as the field expands, as the ability to write DNA increases, as Andrew pointed out, then I think this will have to be constantly reviewed, because the ability to synthesize whole genomes at ease from bottom up, from scratch, will happen. And therefore, one will have to start thinking about the safety regulatory frameworks that would allow those kind of projects to be assessed, because one would then be moving into synthesizing organisms that do not exist in the natural world. And therefore, one would have to think about the implications of that. So this is a really important area of development and discussion. And there are many regulatory authorities around the world that have large amounts of existing paperwork on biosecurity, but also it's a very active area of debate and narrative that needs to be followed through transparently and openly with everyone. But
okay.
Yeah, but for biotechnology applications, I think it's it's not reached that level yet.
One last question, and we only have 1 minute, but
sorry. Okay.
we can use some of the extra time. Yes, this is coming from South America, and they ask, how can we get involved in the BioFoundry movement? How can we learn more? And what would be a guesstimate of the investiture or the cost of a biofoundry?
So one of the great things that there's a working group in the GBA are very interested in exploring how the technology can be diffused into parts of the world that are building this capability. You can start on very inexpensive robotic platforms. So biofoundries is a way of thinking. It's a way of how you integrate your thinking around the synthetic biology design cycle. There are some beautiful robotic platforms from a company called Opentrons in the United States that cost less than $6,000. Now, even buying a couple of Opentrons gets you into thinking about programmability of high throughput assays, high throughput developments, and then integrating existing methodologies that you might have around your universities into this kind of biofoundry concept is the way to start when resources are limited. And then, of course, there are a lot of software tools available. And the beauty about Opentrons is that all of the protocols and all the scripts for programmability of the robotic liquid handling system are available on the web and shared and open source. So there are ways to get in, but I think firstly it's, and actually in South America, there are people very interested in Chile particularly, and also I believe in Argentina and other parts of South America that are actually thinking of biofoundry.
I want to thank the audience for joining us today. I want to thank the three of you, and I want to end our time giving you the opportunity to say your final words.
Thanks, really lovely hearing all of those presentations, and good luck with everything.
Thank you, Paul. Thank you, and I hope the three of you visit Navarra soon. Goodbye.
Yeah, please.